I want to back up a little bit. You're like right.
I'm right on camera.
Yeah.
Oh, that's the camera.
Yes.
Okay. Good. Yeah. All right. Thanks everybody for coming here today. It is great to see you all in person, and I am really excited for Aaron to be giving his final presentation with us. Yeah, Aaron, he is a native of the Chicago area and came to Minnesota with his at Carleton College, got his degree in geology, and we were really lucky to be referred to Aaron at an extension event that he went to talking about soil health. And while at Carleton, Aaron was already involved in undergraduate research. Aaron's already a published author, which is very cool. And we are super excited to see his analyses on nitrogen today. You know, Aaron's been really a flexible member of our team. Like the project that he signed up to do this for is one of those that got postponed and postponed and postponed due to the pandemic. So we found something that was right up Aaron's alley. I like to think, or I hope so. Hope we found something that you enjoyed. And you know, one of the things I would say about Aaron, Aaron in our lab group, I would say, is the most likely to go to the emergency room with an avocado-related injury. Aaron loves cooking, as you could tell with the you know, coffee cake in the back, help yourself. And I don't know. And now Aaron is starting his job at Houston Engineering, which, you know, as my degree was in engineering, I am proud that Aaron got a job at an engineering firm as an environmental scientist. That's right on. So anyway, I will, without further ado, I will turn it over to you and hope you enjoyed the presentation.
Yeah. Thank you, Lindsay. Really appreciate that introduction. I'm really excited to be here today and to talk about my work. It's been a crazy 2 years and it's been really amazing. So I'm really excited to share my work. And yeah, so my talk today is titled Evaluating Nitrogen Losses, Soil Physical Properties, and Biological Indicators in Artificially Drained Fields in Northwest Minnesota. And I'm going to try and talk slowly. I've talked really fast my entire life, so I'm going to really try and talk slowly because I timed it. So if I talk slowly, it'll be the right amount of time. So yeah. Oh. So here's a little bit about the outline of my talk. Let me see if I can do this. Okay, yes. So I'm going to first start, and I'm going to move this mouse out of the way. Hopefully enough out of the way. So first, I'm going to start with an introduction to Northwest Minnesota climate and soils. Then I'm going to talk about chapter 1 of my thesis, which is about nitrogen cycling in a recently drained experimental site in Crookston, Minnesota. And then chapter 2 is about drainage age and its impact on soil physical properties and biological indicators. And finally, I'm going to wrap up with some conclusions and some future work and just some thoughts for the future. So let's start by talking a little bit about Northwest Minnesota and its climate. So here is a map of Minnesota with the mean annual temperature in degrees Fahrenheit, regretfully, but up there circled in yellow is Polk County and Crookston. And as you can see, based on kind of southeast Minnesota where we are located, it's about 10 degrees, a little bit less, 10 degrees Fahrenheit cooler mean annual temperature. So keep that in mind for the context, as well as receiving less precipitation up there. So once again, in inches, but about 15 inches less of precipitation, excuse me, up in Northwest Minnesota over the course of the year. So those are both the context to keep in, and just generally to understand that Northwest Minnesota is cold and receives little precipitation in comparison to Southeast Minnesota. So yeah. And so now a little bit about the soils. So there on the right-hand side there, you can see the footprint of Glacial Lake Agassiz and Crookston in in that red dot. It looks like a metropolitan area there. Yeah, so it's located where in the former Glacial Lake Agassiz. So clay sediments that were kind of at the bottom of Glacial Lake Agassiz, when that receded, those were kind of the parent material and the forming of the soils that I'm working with. So these are really clay-rich soils. And so they have high water holding capacity. They're really clay rich. And so because they are clay rich, they require drainage. And so here is a map. This is using Surgo data, looking at drainage that is potentially benefited and benefited, I guess, absolutely benefited by drainage. And so as you can see up in Northwest Minnesota, it's a pretty dense area of pretty dense cluster of soils that really can be benefited by drainage. even, in comparing the southern part of the state, which we, maybe more closely associate with drainage, it really is kind of a lot of area up there that does require drainage. And so because in future climate projections, we're expected to experience irregular and higher intensity rainfall events, more farmers are turning to drainage in Northwest Minnesota, particularly as it also warms up. More farmers are planting corn, and the amount of acres going to drainage are certainly increasing in Northwest Minnesota. So, part of the reason why Lindsay was hired to be up there is to learn more about what drainage looks like in Northwest Minnesota. There's not a ton of research in this area, but it is expanding, and so that's kind of some of the motivation for this project. And so one thing that I think is important is that I like to talk about the fact that drainage is a management change. I think when we talk about management, I guess we're always talking about things like nutrient application or tillage or something in that realm, something that a decision that a grower makes every year. And drainage is not a decision that a grower makes every year, but it is like a big management change that really has impacts on the soil, on soil properties, on nutrient management, and on the soil ecosystem. So I think it's important to kind of think about it as a management change. And so as more farmers are installing it, we should think about it like it's really going to have an impact that we need to study and kind of think about. So what happens after we install it? This is, I guess, why we study it, right? So what happens after we install it? And so here are kind of three ideas that I'm kind of playing with today that are kind of separate, but also all impacted by being a part of the soil ecosystem. And so the three parts are nutrient management, soil physical properties, and biological indicators. And while these ideas are separate, they are related, and I think they need to be better understood in drainage systems. Drainage research, although robust, kind of focuses on certain areas, and I think kind of opening up into more areas, more emerging realms will kind of provide us more information to growers. So today in chapter 1, I'm going to talk about nutrient management, specifically nitrogen, although phosphorus is very important, but I'm going to talk about nitrogen today, and I'll get a little bit more into that. And then in chapter 2, I'm going to talk about soil physical properties and biological indicators and their connection. All right, so chapter 1 is nitrogen cycling in recently drained fields. Am I talking too fast or am I okay? Good. All right, great. All right, chapter 1. So here's the motivation behind the study. And as Lindsay mentioned, kind of a weird year when I joined on. I joined our lab in June of 2020. And I was originally going to be doing a cover cropping and sugar beet systems. That was originally what my project was. And then due to delays, I ended up getting kind of swooped into this project. So I'm very thankful for Anna and Lindsay for being flexible and kind of letting me go. And after I spent the entire summer working on what was Kyle's project, I joined in. Kyle graciously gave me nitrogen, so I appreciate that. He wanted carbon all to himself, and that was fine. But so after spending the year up in Crookston, I was like, all right, let's do it. So the first thing I want to know is better understand how drainage impacts nitrogen cycling. So there's a really good really robust understanding of course, of like nitrogen leaving the system through tile drains and nutrient loading. But I was curious if we can get a really kind of detailed look at nitrogen cycling specifically and how drainage impacts that. And then also I wanted to quantify nitrogen losses into the atmosphere and waterways. One thing that we talk about, or that is talked about in drainage, is kind of this trade-off, right? So if we're losing more water or losing more nitrogen in our waterways, maybe we are reducing our greenhouse gas emissions. So reducing that. So I want to quantify those and kind of see how that compares. And I want to provide valuable information to growers who plan to install drainage, right? This is why we do research. I'm not necessarily going to be giving any advice today, but I think, right, my research is meant to be built upon, and so that we can hopefully provide more information to growers who are looking to install tile drains, specifically in Northwest Minnesota. So here is a little bit of background context. Crookston, once again, located or marked in red there, and here's a little bit of Canada. And so unlike most of the state, where the water flows down to the Gulf of Mexico, in Crookston, water flows northward to Lake Winnipeg. And so I like to joke that that my research is international affairs because our water is flowing up to Canada. And so like we are partially responsible for eutrophication in Lake Winnipeg. And phosphorus is a limiting nutrient there, but nitrogen controls the toxicity of those algal blooms in Lake Winnipeg. So I think it's worthwhile thing to study and to be thinking about of how can we manage our systems better to reduce our loads in the waterways. And so here are the research questions, which kind of go hand in hand with some of that motivation. First, what are the environmental impacts associated with drainage? So gaseous and also in our waterways. And how does nitrogen cycling change when drainage is installed? So next, I'm going to talk a little bit about the experimental design. And if you were at Kyle's talk yesterday, you saw this image, but flipped. We chose different color schemes for different reasons. But here is our 60-acre field. And what's important to note about this is that we're really going for production scale, right? So Lindsay set these up, right, in order to kind of best provide information to growers. The idea was to make these plots as big as possible. And so the 60-acre field is divided into 4 plots, plots 1, 2, 3, and 4. Plots 1 and 3 are drained as indicated by those kind of dashed lines. The spacing is 50-foot or 15-meter spacing and at about 1 meter deep. And the tile lines drain from the south to the north. And there are also surface drains. So surface drains drain all 4 of the plots. So when I'm talking about when I'm talking about the drain plots, I'm talking about just 1 and 3, but the surface runoff comes from all 4 of the plots. And they all drain to those 2 red dots in the northwest corner of the field. Those 2 red dots are the 2 lift stations, which you will see a photo of in a second. That's where the water collects and then pumps into this ditch, which runs along the west side of the field. And also just a little information, this field has mostly been in wheat and soybeans, but also a little bit of sugar beets. And like there was 1 or 2 years of corn in the last 2 decades. So that's kind of the context of the agronomics. All right, so here's the project timeline. And it's a little busy, so bear with me here. So here is the kind of the extent of the project. It started in fall of 2019 and ended. Will end in 40 years or 100 years, who knows? But for my project, it ends in spring of 2022. And so we had a soybean growing season and we had a wheat growing season in the middle of that. And so drainage was installed in fall of 2019. And so that goes right there. So I guess a little bit off my timeline, but right there, kind of at the beginning. And then the lift stations are turned on in spring of 2020. And so there's the 2 lift stations there. The far one drains the surface plots, and the closer one to us drains the plots 1 and 3, the tile drains. Yeah. And so there's the lift stations getting turned on right there, kind of at the beginning of the growing season. All right, and so now I'm going to kind of walk through the different in the timeline. I'm going to walk through kind of the different assays, the different sampling that we did. So I'm going to start with soil sampling. And so here is us with our Giddings sampler, where we were sampling down to 90 centimeters. So we're doing a 90 centimeter core on each acre. So as I mentioned, there was a 60 acre field, tiny bit less than an acre. We had 64 points, including, so we had like 16 in each of the 4 plots. And so we sampled at each in each acre, and we sampled every season. And so we sampled 8 times throughout the growing season. And those red arrows, so each arrow represents, here, I'll just go back one actually. Each arrow represents a soil sampling. This soil sampling, that one on the far right, happened last week. So it was really late getting into the fields in Northwest Minnesota because of high water levels. And yeah, but so there were 8 soil sampling periods. For my first assay, water tractable organic nitrogen, it was the first 7. So there is 2 before kind of the drainage was turned on, and then we have 5 afterwards. And so we sampled in the fall after harvest, we sampled in the spring prior to planting, and we sampled in the summer kind of during in the standing crops. So that was kind of our 3 times a year. And so water tractable organic nitrogen is a part of the Haney test. It is essentially what we do here is we take a little soil, we put it in water, we shake it, centrifuge it, and then we filter it, and we take that water and we measure the organic nitrogen in it. And so it's telling us what type of microbial foods are available, or what foods are available for microbes. It's kind of a proxy for that. And so that is one thing that we did. And then the next thing is soil nitrate. So we did soil nitrate before each growing season. So once again, I'll be showing data that I received yesterday from last week's soil sampling. And so we did this. And so what's important to note, and I'll remind you of this, is that the soil nitrate measurements are showing you the effect of last growing season, essentially. So we have one prior to drainage, and then we have two after the 2 years of drainage, after the 2 growing seasons. All right, and now I'm zooming in on a growing season. So this happened, all this stuff that I'm talking about happened in both growing seasons, but I'm zooming in on one if you'll work with me on that, if that makes sense. So this is planting 2020 and 2021, and that's harvest 2020 and 2021. And there we had mineralization cores going every other week. So about 10 to 15 mineralization cores. And so that was happening during the growing season. And so mineralization cores, we're obviously taking a look at how the transformations between organic and inorganic nitrogen was occurring. So yeah. And so that is all the soil. And now I'm going to turn to greenhouse gases. This photo is weirdly cropped. I should have fixed this, but you can hopefully see the static chambers that Heidi is kneeling down at. And so we use the static chamber method to collect greenhouse gases every week during the growing season. Well, I guess I jumped the gun on the timeline there. But we had these 5 static chambers, and we did this. We had these 5 set up on each plot. So 20 chambers in total, 5 on each plot, 2 in the undrained, 2 in the drained. And we collected gases over a 40-minute period, and we collected those every week. And so I'm interested in nitrous, and now we get the other half of the photo. We did nitrous oxide for every single week during the growing season, a little bit before and a little bit after. Hope that makes sense. Okay. And so statistical analysis for soil and gas. And so I was using our statistical software. And here I'm comparing drained versus undrained. So I'm looking at the impacts of drained versus undrained in all those assays I've talked about. And I used a mixed model of repeated measures. And I am not going to get into all the variables, but happy to talk about them. And then an alpha of 0.05. All right, so now water samples. So here's these lift stations, which apparently was the only thing I took photos of during my thesis. But here are the lift stations, and I'm gonna talk about water sampling. So here's once again a zoom in on that growing season. So this happened during both growing seasons, but I'm zooming in on the growing season, and we collected every day. So I didn't count if that was actually it, but that was as many arrows as I could put on the slide, and we collected water samples daily. So luckily, we were not collecting by hand. We had these auto samplers that were kind of located. I'll just go back to them. There's auto samplers, which are located kind of sitting on top of the lift stations there. And so those are collecting water from inside the lift station. So it's not necessarily collecting the water that is leaving, but it's what's in the lift station and kind of representative of what is pumping out. And there we have total nitrogen, nitrate, and ammonium and being measured from those water samples. And so those water samples are collected 200 milliliters every 6 hours. And so into a composite 1-day sample. So the 1-day sample is homogenized with every 6 hours throughout the day. So each sample represents 1 day. We also measured discharge. So we used pressure gauges to measure to calculate based on the water level to calculate how much water was leaving the system in each pumping event. All right, that was the timeline. I hope that was not too bad. Statistical analysis for water samples, once again, R. And here I'm comparing surface and subsurface and drained and undrained. So as I talked about earlier, right, drained is plots 1 and 3, but undrained includes the surface plots from all 4. So I'm doing both. And I had non-parametric data here with an alpha of 0.05. And other things we measured yearly or new yearly, right? So we measured yield as well. This wasn't an agronomic study, and so I wasn't specifically, we weren't out measuring yield. We were just this is just what came off the John Deere. But we also knew the nutrient inputs. So yeah. All right. And now a little context on the precipitation. All right. So here is the 30-year normal for precipitation over the course of the year. And so also I wanted to point out that because of kind of other studies, I didn't treat my study using water years. I used the calendar year because of how frequently the ground is frozen in Crookston. So I decided to analyze based on the growing seasons and the calendar years and not on the water years. So hopefully that'll make sense later why. But basically, as you can see here by the normal, we had the highest precipitation kind of May through September. But as we know, 2020 and 2021, not normal years. 2020, we had pretty excessive precipitation, we had high precipitation, mostly in July. July had about like double the normal amount, and then pretty low in the fall. And so just a reminder to keep in mind, I'm going to keep reminding you about the precipitation, but the precipitation is a big story because we're talking about drainage. So just keep that in mind, really high precipitation, specifically right in the middle of the growing season there. And then in yellow, we see 2021. And so here we had a crazy low amount of precipitation. I don't remember it raining at all in July in Crookston. But then as we got into the fall, we had a little bit higher precipitation. September and October were specifically pretty high in precipitation. But that was, of course, like after the growing season. The wheat was harvested in August. So yes, all right, nitrogen cycle. I'm gonna take some water before I talk about the nitrogen cycle. Looks like my timing is going okay. All right, so here is the nitrogen cycle. And so the reason I'm kind of base everything in the nitrogen cycle is because the nitrogen cycle can help us understand where nitrogen is moving. It's a really useful tool if we're able to capture how it's moving. We can kind of understand how moisture interacts with nitrogen transformations and losses. And so the nitrogen cycle, you can break it down in a number of different ways. This is the way I'm breaking it down. There's losses, right? So there's crop uptake. When we harvest, we lose nitrogen through that grain. There's denitrification, there's gaseous losses. And then there's leaching and runoff. So leaching is the equivalent of losing through the tile drains, and runoff is the equivalent of losing through the surface drains. So these are losses that we are measuring. There's transformation. So within the soil, there are transformations, right? There's mineralization and immobilization, ammonification, nitrification, the movement between organic and inorganic forms of nitrogen. And there's kind of more stable or known pools. So we know how much nitrogen we were putting on the fields, and we can measure kind of some of the organic pools. So I'm not measuring everything here, but what we can do is we can do a lot of comparisons between the drained and the undrained to kind of better understand how the effect of installing tile drainage, or what the effect of installing tile drainage has on nitrogen. All right, so let's start with soil nitrogen transformations. And just to like I'm going to be switching a little bit back and forth between drained and undrained, and what's the other comparison? Drained and undrained versus surface versus subsurface. So right now we're just focusing on drained versus undrained. And so first I'm going to talk about these transformations. And so I'm going to start with 2021 mineralization, and I'll show you why I'm going there first. And so we had a drought, remember, in this year. And what's interesting here is that we had in the drain section, we had increased mineralization right at the beginning of 2021. And so this is kind of logical because right after nutrient application, we still had some moisture in the profile, and we saw that increased mineralization due to kind of the moisture and then the aeration. We had some mineralization in the drain, but then we had negative or immobilization kind of throughout the rest of the growing season. This has been kind of seen in some papers when we do have drought conditions, that we do see negative mineralization. So that's kind of interesting. But what's important is that in that drain section, we had that really, what we should be seeing, right, is that that mineralization right at the beginning of the growing season from kind of the impact of the tile drains. And so sadly, I don't have 2020 mineralization data, but what I do have is we're going to be able to kind of piece the story together through other ways. So come back to that. I'm expecting that we saw something similar to this. I'm expecting that we did see, we probably saw positive mineralization, but much more in the drain section than the undrained. But I'll show you why I think that later. All right, next let's talk about nitrate. And so I want to keep in mind, I'm talking about soil nitrate prior to planting. So the years here don't match up with the growing seasons. They match up with the year they were sampled. They were sampled in the spring. So I hope this will make sense. So 2020, spring of 2020, this is before drainage was turned on. So this is prior to having drainage, and this is from the effect from the 2019 growing season when there was no drainage, right? So we basically see no difference between the drained and the undrained plots. This makes sense. There's no drainage effect, so it makes sense that we wouldn't be seeing any differences there. But then we get to 2021, and we see a different story. We see, remember, 2021 had a very high precipitation. I'm annoyed that that's cut off, but in 2021 we had high precipitation, and we see a depletion of nitrate from the drain section, particularly in the upper 60 centimeters, right? So we had high precipitation, which means we were depleting the nitrate levels. There's a number of different pathways that we're going to talk about for where that could have gone, but basically we're seeing less nitrate in the upper portion of the soil profile, but not down low. And so that is the effect of the 2020 growing season, the high precipitation year. And then in 2022, this is from the low precipitation year. And I'm not even kidding when I plotted this yesterday. I received this data yesterday. So the interpretation, we're working on it. But so this is from the low precipitation year. And we once again see that depletion in the upper 30 centimeters. But what's interesting here is that in the drain section, in the lower 60 centimeters, we actually see higher nitrate levels, and particularly very high, in from 60 to 90. And this is likely, Lindsay told me that they were hitting water with the probe often, and this is probably because we had pretty low precipitation in 2021 growing season, and so likely we weren't losing as much of the nitrate through the tile drains, but instead it may have been holding in the water kind of lower. The macropores were able to take some of the water down and kind of settle that nitrate in the lower portions of the soil profile. So hope that story makes sense. And let's talk a little about crop uptake. And so I don't have crop uptake data from both years, but what I do have is a good proxy, which is, or at least what I'm going to use as a proxy, which is yield. And so shift your mind back to now to the 2019, 2020, 2021 profile. So 2019 didn't have drainage. And so as you can see, pretty similar yields between the drain and the undrained because no treatment there. Then in 2020, we had the high precipitation year. And we see the impact of the drain, that we see the drainage working, right? We see that we have the higher yield in the drain section due to, just all the reasons why drainage kind of helps increase yield, including higher plant uptake. And so when we saw that depleted nitrate in the upper 30, 60 centimeters, presumably some of that was being taken up by the plants. And so that's why we see higher yields and lower nitrate. And then in 2021, we had a water limited year. So this is interesting, right? Because water was limited, we probably had pretty reduced plant uptake. And in our plant uptake measurements, we saw higher in the undrained. And that is probably leading to that higher yield. So because the undrained had a little bit higher moisture because it wasn't draining as much, we saw higher yields there. So hopefully that all kind of connects together. And then I'm going to talk a little bit about organic matter now. And this is kind of a more stable pool, and it's very stable. So this isn't soil like organic nitrogen in total. This is the one that we decided to look at because it's part of the Haney test, and we saw this is pretty stable throughout. This is much more kind of tied to seasonal variations rather than to drainage treatments. And so, yeah. And so now I'm going to kind of sum up soil nitrogen transformations. We saw increased mineralization in the drain section, and I'm assuming in 2020 as well. And when we had high precipitation, we saw soil nitrate was depleted in the drainage in the drain sections from the upper 60 centimeters. And in low precipitation, it was depleted from the upper 30, but enriched in the lower part of the soil profile. And plant uptake might account for some of that, at least some of those differences, particularly being seen in 20 when we had the high precipitation year. But what about other loss pathways, right? There might be other things that are happening. It's not just plant uptake. And so now I'm going to talk about the big forms of losses, including greenhouse gases. So often, as I already mentioned, one of the big viewed as one of the big advantages of intensification of drainage is that we're going to hopefully have reduced greenhouse gas emissions, reduced nitrous oxide emissions in those drain plots due to less saturation of the soil profile. And so, here's denitrification back on the nitrogen cycle. And what we saw was exactly what we expected to see. And so there's a little precipitation graph that I'm kind of going to put a lot everywhere, but just to remind you about what was happening in 2020, this is 2020. We have cumulative nitrous oxide, and we saw that the undrained section had greater emissions over the course of the growing season. This checks out with what we're seeing. This makes a lot of sense. And 2021, we saw something similar, right? We had lower precipitation, but we still had higher in the undrained section. But what's really interesting is we had this huge emissions event right at the beginning of the year. This is likely coming right after nutrient application. We have that huge emissions. We still have a little bit of moisture in the soil profile from the previous year. and just from a probably a high water table coming out of winter. But then those kind of level off as we had pretty low moisture. It didn't even really increase much when we finally did get some moisture in September and October. So yeah, so that is greenhouse gases, and now I'm going to turn to edge of field losses. Edge of field losses. So I'm going to start with moisture. So a sanity check to make sure our drains are working is just looking at moisture. As you can see in 2020 in the top there, and I put hopefully correctly where the lift stations are turned on, we see that the undrained section kind of separates a little bit from the drained section, has a little bit higher moisture, which just basically means our drainage is working. We're having a little bit higher moisture content in the undrained section. And then in 2021, we see it coming out of the fall, or out of, the winter. We see kind of the higher moisture, but then things kind of stabilize as we're hitting those drought conditions. It's much more controlled by kind of the drought there than kind of the treatments. So that's just something to keep in mind as we're looking at this data. And so now let's take a look at the losses, right? So runoff is our surface losses, leaching is our subsurface losses. And so now it's like turn your brain, I guess, from drained and undrained to now subsurface versus surface, and then I'll kind of tie it back together. And so here's the color scheme I use. It's the greenish is the surface, the green blue, and then the kind of lighter blue is the subsurface. And so just as a reminder, the surface is draining all 4 of the plots, and the subsurface is draining just plots 1 and 3. Okay, so discharge. Once again, here's the precipitation. And so discharge is low. And so that's kind of a part of our experimental design. I showed you those big lift stations. They're huge. They hold 600 up to 600 gallons of water. And so our discharge is just generally lower because it pumps out only when it reaches a certain level. That depends, that kind of varies. But discharge is generally lower than other studies. But what we do see is it really follows precipitation closely, right? So we had that high precipitation in July and August. And we see kind of a really big increase specifically from the subsurface drains. The surface drains are a little bit more steady and certainly level off when we have low precipitation. And then check out the scale here. So the scales are about an order of magnitude different because 2021 was very low precipitation. And October, we don't see any we don't see very much discharge until October when we finally see some out of the subsurface. So very low discharge in 2021 compared to 2020. Okay, so let's talk about loads real quick. Precipitation one more time. There'll be a quiz. All right, and so first I'm gonna talk about nitrate loads. And so in 2020, once again, the word precipitation doesn't seem real to me right now. I've said it probably 100 times in the last 10 minutes, but precipitation, nitrate loads are really following precipitation closely. One thing that's important that kind of happened in our study is that we really never had concentrations above 10 ppm. They were relatively low concentrations, and they didn't vary all that much. Well, they did vary some, and I can show those, but didn't vary like wildly during different points of the growing season. And so here, as you can see, the subsurface had the highest yields right in July and August when we had that high precipitation. And then kind of leveled off a little bit more as we went. The loads were always higher in the subsurface than the surface drains throughout the growing season. And then in 2021, we saw really decreased loads of nitrate until we get to October. So there, and the subsurface loads are higher in October than the surface in October. And total nitrogen. So our subsurface total nitrogen loads also are higher, and they are once again moving with precipitation. So this looks very similar to the nitrate. Once again here, they are following precipitation pretty closely. So yeah. And so now let's talk a little about drained versus undrained. And so remember those plots, it's drained is only is coming from 1 and 3, and undrained is coming from all 4. But the subsurface loads were much greater than the surface loads. And so now if we plot, so what I've plotted here was every single load event. So every daily load event is on the y-axis and then broken out by month. So what I have here is every total nitrogen, every nitrate. And so in yellow is the drained and in blue is the undrained. And so what we see here is particularly during the really, really high precipitation periods, we see much higher loads in the drained section than the undrained section. This kind of intuitively makes sense, but it really is kind of much bigger in the drained than the undrained. But then when we get to 2021, low precipitation, we see basically no difference until October. This is really interesting because basically in a low precipitation year, we're not going to see, the drains are not going to be pumping. And so we're going to see very similar losses between the drained and the undrained. Yeah. And so what are the differences in losses between the 2 years? So if we can think about nitrogen, this isn't really a budget, but this is just thinking about our losses kind of in context. And so 2020 losses, the drained are in yellow there, are much greater than the undrained. And this is just looking at nitrous oxide and then losses from surface and tile. As you can see there, it's about 40 kilograms per hectare larger in the drained portion. So that's a fair amount of nitrogen. And that was a significant portion of that is coming from the subsurface drains. But then we get to 2021, we see that they're actually pretty similar. I mean, they're still 2 kilograms per hectare different, but think about that, 40 versus 2, it's a big difference in terms of how much we're losing during a high precipitation and a low precipitation year. So here's kind of the conclusions from chapter 1 is that increased mineralization and decreases in nitrate particularly occurred in 2020 and probably in late 2021. In the undrained section, greenhouse gas losses are increased, but at a much smaller magnitude than kind of the sizable losses that are seen from the drained sections through those subsurface drains. All right, chapter 2. Doing on time. All right. I'm going to have to talk quick now. This section is a little bit shorter. So this section is about long-term drainage. And so this project kind of came about in a different way. We applied for some extra money and we were looking to kind of think a little bit more about, Lindsay has these amazing plots up in Crookston. What are they going to look like in 20, 30 years? What things might be helpful to look at? And so we decided that we wanted to develop a study that kind of can be a 1-year observational study where we can learn a little bit about how drainage age impacts soil properties. And what I mean by drainage age is how long a drainage system has been in the ground. And so I'll note long-term drainage studies that kind of focus on like long-term plots, kind of focus mostly on discharge and nutrient loads. They focus on what's leaving the system, which I just talked about for 20 minutes, right? So like what's leaving the system. And they hypothesize that basically the discharge and the nutrient loads get greater as time goes on, and that these preferential flow paths are kind of these places where nutrients can quickly move through the soil profile to the tile drains are a potential cause for kind of these increased discharge and increased loads from tile drains. But none really examine the soil properties. A lot of them hypothesize, but they don't really examine the soil properties. So we thought, can we design a 1-year study to help guide some future long-term trials and maybe kind of take a look at how are soil properties changing over time? And obviously I don't have an entire career to do that. So I was like, let's just do it in 1 year. So the best way we could do that, well, I'll start with the research questions first. So does drainage age impact soil physical properties? So that's one thing we're looking at, right? Because we saw that these preferential flow paths were being hypothesized as developing. Can we identify those? Does it impact biological indicators? So kind of some soil health metrics, are those being impacted by drainage? And what mechanisms are causing them and are they connected? And so the way that we thought to best answer this was to create an on-farm study to examine drainage age and its impacts on some of these properties. And so we were located up in the Red River Basin and we had to select some sites. And so we wanted to choose some sites that had standing wheat or standing corn, want to keep kind of the nutrient application things separate. And we had drainage installed. We wanted for our drainage to be installed during periods of interest. So we were looking to find some older drains and some new drains and kind of compare them. And our textures ranged from loam to kind of clay. I was trying to really narrow it down based on Surgo, but you get out there and it's not what Surgo tells you always, and that's okay. But yeah, so here is the map of our sites. And the reason we went all the way down here is because it was very difficult to find people who drained in the last 5 years that were planting corn that wanted to work with us. So that's why we were kind of driving all around. Thank you, Kyle. Yeah, so here's a little timeline, and I started with the year I was born to put it in context, and to the year of, I don't know, my hopefully master's degree. And so here are 3 farms that we found that were drained kind of right at around the turn of the century. and 2 of them are located in Crookston on the NW Rock campus, and 1 is a on-farm site. And then we found 3 that were drained kind of in the last 5 years. And then we had 1 that was undrained. And so I will refer to these as the greater than 15 year, and that represents they were drained greater than 15 years ago, less than 5 fields, drained in the last 5 years, and 1 undrained. And so the undrained they're not really a part of the statistical analysis because don't really have a lot of robustness there, but just as kind of a I don't know, a gauging point to kind of look at. Yeah, and so here is a table. I swore I would never put a table in a presentation, but I will come back to it if people are interested in kind of the soil properties and stuff. So yes. All right. So remember, these are drought conditions. And so yeah, it's there. So keep that in mind. That's an important context, right? It's one thing that we weren't looking at, and thankfully we weren't looking at, was that we weren't looking at the loads and the discharge because there was none during July. So it would have been tough to look at it because we would have been waiting by the tile drains all year. So yeah, that's drought conditions, and that kind of informs some things, you know. Right, if we had multiple years, maybe we would have chosen to not necessarily sample in July, but this is kind of how it worked out. I didn't know when we started sampling that it wasn't going to rain for a month. So yeah, so let's start with the soil physical properties. And so the first kind of most important one, or not necessarily most important, but something that we were trying to kind of corroborate with other studies, was how quick is water moving through the soil profile. And so we have this beautiful dual head infiltrometer that kind of, when it works, does everything for you, which is really nice. And it measures how quickly water is moving through the soil profile. That's saturated hydraulic conductivity. And then next we did bulk density, and I was using kind of this hammer core. I broke one of these because it was so dry. And you can see a really nice compacted one on the right-hand side there. So I was working with compacted soils. One day I couldn't even get down to 30 centimeters because it was so dry. Or I should say Kyle. Kyle did a lot of the soil sampling. I appreciate it. And we sampled 2 depths, so 0 to 15 and 15 to 30. And once again, I don't think we could have gone any deeper, seriously. It was so dry. And then also, let's see if this video works. We also did, oh, wow. Okay. You weren't as excited about the video, but aggregate stability, which is essentially we're taking kind of like a chunk of soil, or chunk, that's a terrible term, but we're taking an amount of soil, and then we are kind of sieving it in water to kind of see how stable those aggregates are. And yeah. Oh, uh-oh. All right, biological indicators. And I'll get that out of here. Biological indicators, and so you chose 3 here because they are, one, we are doing them out on field 5, or not field 5, excuse me, on the chapter 1 site. We are doing these things, and so we thought it'd be interesting to compare. Also, they're part of the Haney test, and so we thought that in sake of standardizing soil health metrics, these are the 3 that we chose. Potentially mineralizable carbon, or PMC, is also like respiration. Is basically we incubate soil and measure the amount of and wet it, and then measure the amount of carbon dioxide that comes off in 24 hours. That is measuring how active kind of your microbial communities are. And then we have water extractable nitrogen and carbon, which are, once again, measurements of kind of like the microbial food. And once again, those are formed from extracts. And also 0 to 15 and 15 to 30 centimeters. So models, here's my mixed model. And there's the level of significance, 0.1. We increased it because it was a field study. So yes. Yeah, so there are other things that I was potentially trying to look at, which was distance to tile line, but they ended up kind of not working out as well because it was hard to find the tile lines on these farmers' fields. So it's kind of hard to do, but yeah. So let's take a look at some of the results: soil physical properties and saturated hydraulic conductivity. And so here is the difference. And in blue, we have the greater than 15. In yellow, orange, we have less than 5, and the undrained there. And as you can see, there's a big difference between the greater than 15, less than 5. So this is kind of corroborating other studies. We see an increase of about 7.6 centimeters an hour of water movement, which is about 3 inches, which is a lot if you're thinking about a big storm event. So that is pretty interesting there. And bulk density. So we did see a decrease kind of at the top in the 0 to 15, but mostly at depth we saw a decrease in bulk density. Developing of kind of those preferential flow paths and those macropores would hopefully decrease bulk density. Bulk density, just like caveat, bulk density and drainage is like really kind of most people think it like, or most studies have kind of found that it decreases, but there's also been tons of studies that show an increase. I mean, bulk density is kind of fickle, and we're also sampling under dry conditions. So bulk density is kind of a tough one to kind of parse out. And then aggregate stability. And so this graph, I know I'm hitting you all with a new color scheme. I guess it's a similar color scheme, but it's a new type of graph. And so this is the proportion of the sample. So after we sift it, the proportion of the samples that were left. And so the bigger your light blue section are, the more stable it is, right? So this is the biggest, and these are the smallest fractions. These are your macro aggregates. So the bigger they are, that means the more stable kind of your aggregates were. And so as you can see, there's not really a huge difference between the greater than 15 and the less than 5. You know, the levels of macro aggregates are pretty close. So that we were kind of like, didn't really see anything there. We were kind of a little bit confused. And so as I was looking at the data, I realized that tillage is really kind of the controlling factor here. We know tillage controls aggregate stability, or at least is a big factor. Farm 2 was the only, which is a less than 5 field, is the only field with reduced tillage in kind of this in this experiment. And so that had really, really high aggregate stability. As you can see, almost like 90% of the sample was basically those big macro aggregates. And so that was kind of controlling the aggregates, and not necessarily our drainage treatment. All right, biological indicators. All right, so PMC increases with drainage age. So as you can see here, the greater than 15 in blue is higher, particularly at depth, in kind of, in those kind of in those fields that were drained a longer time ago. And so that's pretty interesting. And we're also seeing it in kind of our water soluble organic carbon and nitrogen. We're once again seeing, particularly at depth, and then at both depths for water soluble organic nitrogen, we're once again seeing that it is, these biological indicators are much higher in the, or not much higher, but higher in the greater than 15 field. So this is pretty interesting. But what about other factors, right? So I had some other factors in my model that you saw. And so what were the ones that also kind of were complicating and kind of other also factors? And so one of the other things that was really important was this difference between corn and wheat. And I had a nice kind of try to create like as much symmetry as I could between the data sets. What's interesting here is particularly at depth, the corn had much higher PMC than wheat. And this also was happening in the water-soluble organic nitrogen and the water-soluble organic carbon, right? So at depth, particularly, we saw much higher biological indicators in the corn than the wheat. And so what's going on here? What is happening? And so what's happening is that we have two timescales at play, right? So seasonally, we were sampling in July. The wheat was it was a super dry year. The wheat was kind of on its way out. It was really close to physiological maturity, lower rates of photosynthesis. The biological activity was less than the corn, which was kind of still in July, like peak growing times. And so they might be changing. The biological indicators are changing based on the standing crop in the seasonal. So in kind of the smaller scale. But if we're looking at like kind of the bigger scale and the decadal scale, these preferential flow paths are creating hotspots. And so this is something that's talked about, as we talked about a little bit more recently in the literature. It's that these preferential flow paths act as hotspots where if we re-wet them, we then have these bursts of biological activity. And so this is what we're guessing is happening here is that these preferential flow paths that are developing in these fields over time, like as we're increasing the infiltration, we're increasing these flow paths, and we're increasing the biological activity in those pockets, and that's how we saw that increase in biological activity. And from our observation, it appears that drainage systems are changing soil properties as they age, right? So this is kind of something that was kind of thought, right? Is that we're seeing increases in discharge and nutrient loads, but here it's pretty, from our observational study, we can see that it appears that that the physical properties and biological indicators are changing as drainage systems age. All right, so what are the implications of that? For growers, it means that we can expect faster moving water as the drainage system ages. You're going to expect the infiltration to be quicker, water is going to move through the soil profile, and it's going to bring nutrients with it, right? It's going to increase your nutrient transport. So thinking about that when we think about nutrient stewardship, water is going to be moving quicker through the soil profile. And finally, increased microbial activity with drainage. This is a little bit of I put a question mark there because I couldn't commit to putting that bullet point fully. But I mean, we are seeing that. I don't really necessarily know what the implications are, but it is something to keep thinking about and keep exploring, is that microbial activity is we're seeing it increases as drainage gets old. And so for researchers, what does this mean? It means that we need more long-term drainage trials that look at properties and not simply discharge and loads, right? So obviously there's a lot to be said, like there's a lot to be said, of course, about nutrient loading and why that's important, but there's also other things. We're changing the soil ecosystem, and we need to be kind of thinking about that when we install drainage. And we need to look at how these factors might be changing in different soil types, crops. There's so many different ways that we can kind of think about it. Ours was a very specific study, or a very specific, location and precipitation. So we need to look at it in different ways. And so both chapters in sum, starting with a story. So this is just like a story I want to mention. And so that second chapter was, as I mentioned, an on-farm study. And one of the farmers we ended up working with, as I was calling around to try and find farmers who are going to collaborate with, I talked to this farmer I was explaining the study, and he said to me, he's like, Aaron, why should I let you come out and do drainage research on my field? Someone from university not to be named, not our university, told me to install tile drains, and it's probably the biggest financial mistake I made in my entire time, in my entire agricultural career. He's like, I spent $500,000 drainage, and I get in my field 1 or 2 days earlier. It's not worth it for me. And so, we had it back and forth, and he ended up being an amazing collaborator, because we talked about how, we talked to the project and how I wasn't trying to convince anyone, but I was trying to think about drainage in a better way. And so I think, that what that story reminds me of is that, we're working with growers, we're trying to figure out more information for growers and kind of work with them. And we also need to think about that it is a big management change and a big investment for farmers. So that's just something that I could think is important. And so as we move forward, Northwest Minnesota is going to experience irregular and higher intensity precipitation. That's something that's just going to happen under projected models. And so more farmers are going to utilize drainage to maintain their agricultural productivity. And so with that, we need to realize that drainage is an investment, and it's also a change in management. It's both of those things. And as researchers, we need to better understand these systems and how they impact soil properties and nutrient movement. And these things are essential. So that's kind of the last word that I'll leave you with, except for some future work, is that hopefully out on field, I keep saying field 5, I'm sorry, in chapter 1, we hopefully will do some N-rate trials. So build upon my work to kind of give more specific recommendations to growers. And we can hopefully do some more projects looking at long-term soil physical properties and biological indicators. And both of these things with more rotations, textures, management practices, etc. So so I want to thank Minnesota Corn Growers Association, as well as AFRIC for funding this research. And a huge thank you to Lindsay and Anna. I don't know, I can't say enough about how incredible these last 2 years were. And I guess sorry, I'm not going to cry, but when the funding was gone, or when the funding kind of disappeared in the summer 2020, both of you stuck with me, I guess, and that meant a lot. So and then the extended lab family, all those people, thank you so much for everyone who did a lot of field work with me and ran samples with me. Thank you so much. And then Fabian and Jeff for serving on my committee and their contributions to the study. So thank you so much. And any questions I'll take now. I can get Zoom out. 4 comments in the chat. Oh, all right. Questions? Yes, John.
Aaron, really nice job. Really
Thank you.
Stuff, really interesting. I was wondering, assuming that the results in 2020 are what we could expect most years, right? 2020, we all know was out of the ordinary. You saw increased yields, reduced nitrous oxide emissions, and increased nitrate loss. Subsurface in the drain treatment. So drain versus undrained, what would be better overall for a producer to do? If they get those benefits and we get environmental benefits and yield benefits with the reduced nitrous oxide.
Yeah.
But we do see more loading into Lake Winnipeg. So
yeah.
trying to.
Yeah, I mean, this is, yeah, great. This is like, I guess, the big question. And this is what Lindsay commented on when she was like, Aaron, you need to say something, right? You need to like make like at least say something about like, what would you recommend to a farmer? And I don't know, hard to like commit to something. I mean, I think drainage is going to become important. And I think, right, the combination that we really need to figure out is like, this is why this research is happening, is like, we can install drainage, but have better nutrient stewardship, and we can have the best of both worlds, I think, right? It's like, if we're figuring out better ways to kind of reduce our losses via waterways, but still have drainage, like I think there's a way to do it. And I think it's right, it's investing in more research, it's investing in kind of doing N-rate trials to say, all right, when you're installing drainage, you don't need to, you don't want like this many pounds, right? You need to be thinking about kind of pulling it back a little bit, because you're going to be losing more into your drains and stuff like that. So, I mean, I think realistically, like, more farmers are going to need drainage, especially, in clay soils. And I think what we need to do is kind of invest more and more in kind of doing more N-rate trials, and just kind of thinking more critically about applying the right amount of nitrogen, so we're not losing as much. I know that's not really an amazing answer, but I hope that's fair. I know like a lot of people have opinions on this and stuff. So
That's usually about the best we can do,
right. Yeah. So yeah. Other questions?
Aaron, can I ask you a question?
Oh, yes. All right. Yes.
This is Dr. German from Northern Ohio. My question for you is when I looked at your data, you were comparing the means and the medians of things, but I also noticed that lots of your data had differences in variation. And particularly I noted, for example, slide 65.
I'm going to it. This is my aunt, everyone.
Who actually pays attention to data too much.
65.
I wrote down 65. Try going back the one before this. Let's see. That one. Yes. One more forward. If you look that you've got a big change in variation in things, and often we use differences in variation as the basis of a mechanism for explaining what were happening. Did you think at all about variation in your measures? Go the slide before this one.
Oh, wait. Oh, sorry.
Even.
This one.
Yes. So that you can see in 2021, the variation was a lot less than it was in 2022. Any thoughts on that?
Yeah, so I guess the reason I think like the difference between, I guess, an agricultural study, and maybe this won't answer your question, but like the difference between, I guess, you do research in like kind of in a lab, and I guess there is obviously variables, but a lot of the variation here is also coming from the fact that we're working on a 60-acre field with two very pretty distinct, this is something I didn't really talk about, soil types. And so there is a lot of natural variation in this field regardless, right? Besides the treatments, there was a lot of variation kind of occurring. And so I think that's kind of an important thing to note, is that in a 60-acre field, right, it's so hard, and this is why we have precision agriculture, right? It's super hard to predict how like nutrient levels like within 10 feet of each other, right? Like it's really, really variable. And so like that natural variation is just going to be happening when we're working in like kind of an agricultural context. I feel like I'm not answering I can't see you, so I don't know if I'm answering your question well, but I hope that makes sense. Like, yeah, there is a ton.
That's a great answer.
Yeah, I just think
that is a great answer.
I'm getting embarrassed right now. All right. Thank you, Becca. No, I mean, I think that's what's important to notice, is that like there is a lot of variation in agriculture because there is just so much topography. There is just so much natural soil variation just from soil forming processes and stuff. So yeah. Good. All right. Questions in here? Yes.
Okay, quick question. You talked a little bit about leaching, and you talked about subsurface drainage.
Yeah.
You talked about the precipitation. I'm curious, because there's obviously going to still be leaching in these
Yes.
non-drained spots. And one of the things you had is measure additions of all the last fields, like subsurface with zero undrained spots.
Yes.
But I'm curious, did you measure leaching in any way from the other plots, from undrained plots? And I mean, how would that compare then if you did include some of the
Yeah. We didn't. Sorry, go ahead.
Can you repeat the
Yes, repeat the question. Yes. So Melissa asked, what is obviously, so I kind of, what about leaching below the tile drains? Because the tile drains are only 1 meter deep. So there's obviously going to be some leaching below that also doesn't just go into the tile drains. And so my answer is it's an oversimplification, I guess, is the answer is, we measured kind of, what we thought was important, and we thought we would capture most of it without kind of looking at kind of leaching. I don't know if there was thoughts on that. I don't know how do you measure leaching in like this type of system? How would you do that?
Yes.
Yeah, I'm asking you, Lindsay.
Okay, I wasn't sure if you were looking at me or looking at Melissa.
I'm just curious.
Yeah, I think you need a lysimeter.
Yeah.
Which we didn't put any kind of a lysimeter
Yeah.
out there, but yeah, it was just kind of one of the known confounding factors
Yeah.
that we were gonna have to deal with.
Yes, this zero is an oversimplification. Yeah, but and I think that's what I think is something that I tried to make clear, and maybe I didn't state this enough, right? Is like I'm trying to compare drained and undrained, and I'm not trying to capture every little bit of nitrogen because it's really, really hard. It's almost impossible to capture every little bit of nitrogen, and so the comparisons between the two were the best that we thought we could do. So thank you. All right, thank you.
I had actually the same question that Melissa had.
Okay.
But if you go back to your nitrogen load graph, and I saw on the Y scale, you have is that 1 kilogram nitrogen per
Yeah.
hectare? Why isn't it so low? Because when we do measurement in sandy soils, our nitrate leaching, our N load are in the range of 25 to 30.
Yeah. So the reason it's so low is because of like our experimental design was that basically the pumping events were super, super small, basically. Like the discharge from the entire thing, I hope I don't have like an axis labeled wrong, but like our discharge was so small that essentially our loads were incredibly small and our concentrations were also pretty low.
Yeah, Aaron, can you comment about what level the concentrations were?
Yeah, oh, I can. All right, great. Hidden slides.
We told them to take it out because there were a lot of slides.
Oh, here we go. Here are the concentrations. So here's like total nitrogen concentrations or nitrate concentrations. So they were pretty low, and this is a logarithmic scale. So particularly in 2020, we were basically at like less than 2 ppm, and then that on top of the fact that our discharge was like in the like less than a millimeter for the entire year, because we were storing so much of that water in this storage in kind of those lift stations. Basically, like the pumping events were so super low in terms of loads. Does that?
Yeah, so can you comment, is there maybe a difference in soil type? Because I'm talking about sandy soil and maybe cropping system. Do you have any?
Yeah, I think just generally, right, where I think we're gonna have lower loads just in a clayey soil. I think it's gonna hold on to that nitrate a little bit better. But I mean, I think that's generally, I think there's like a lot of reasons, and clay soils being one of them is kind of the reason why the loads were just super low in the study. And I mean, they're like pretty shockingly low. It's pretty hard to find some that are that low, especially also with the lower precipitation happening in Northwest Minnesota. But we did see some, there are some studies out of Canada that have like similar really low loads, with their lower precipitation as well.
Because I was just looking at the data of my research, and in sandy soils in Westport, which is not very close to Crookston, but almost same precipitation. I got around 100 ppm, like parts per million in few events from lysimeters. So it's not rain, but continuous corn.
Yeah. Also, 2020 was soybean year as well, and then 2021.
Okay.
So 2020 was soybean, and then 2021 was no precipitation. So those two things combined means, the year that we could have had probably much bigger losses. This is a great like advertisement for drainage. It's like it's always gonna be lower than 10 if you're in a soybean year during high precipitation, or low precipitation the year that you're actually applying nitrogen. So
Great.
Other questions? Yes, Les.
One mitigating factor in the Northwest is that a lot of those watersheds have put in storage, trying to mitigate flooding, and you can get the denitrification in that storage as well. So if that's one situation that's different from, say, the Minnesota River Basin, where that stuff goes straight to the river and out the door.
Absolutely.
So I one thing that can mitigate what's going on up there.
And Lindsay's working on some bioreactor work as well. So, yeah. Anything else? Kyle? Oh, a question on Zoom, maybe?
No.
Oh, okay. Okay. Can you repeat the question? Oh, okay, sorry. Okay, is that it? Anyone else? All right, I brought a cake, so we can bring the cake up.
A second cake.
A second cake, yes. All right, and I was told that my partner Abby really wants to see the cutting of the cake. I don't know if she's still on here. She's a school teacher, so she might have.
Do you want me to take a little video? Or?
Yeah, you could. Yeah, I'm trying to figure out if we can do this via here. Oh, there's no candles. Oh. Okay, I'm gonna try to do this genetically.
How do we look on Zoom? Can you look on Zoom?
Well, I want to. Can you? You can't go over to the other side. All right, I'll turn the cake around.
Turn on the lights.
Well, that's what I was trying to work on too with that.
This, I really hope this worked because this might look really dumb if it didn't work. So, I hope. All right. Okay.
Wait for the lights to be on.
I don't know. I don't turn it on up there.
What? All right.
Good job.
All right. And so people have to eat cake, by the way. They don't have to. I'll bring it to the barbecue tonight if not. But hopefully this worked. I'm going to be really.
You're sharing your screen.
Oh, I am.
Yeah.
All right. Can people see it? No.
Yeah.
All right. People can. Okay. All right. Well, I'm just going to cut it and we'll see.
Yeah,
This
there
is
you
good.
go.
I can see it.
Okay.
Yeah.
Okay. Oh, I hope this worked. Oh, God. All right. I'm trying. Let's see if it worked. All right.
What's the bulk density?
Pretty dense. All right. Hopefully this worked. Uh-oh. Oh. It's nice to be tile lines. We see two horizons, a parent material, and two tile lines hidden in there. So hopefully it looks. That looks pretty good.
It does.
All right. Thanks, Abby.
And then you have soybeans and wheat.
I do. I have soybeans and wheat on top. So thank you, everyone online. Thanks for coming. I appreciate it.
Can I help you by cutting this?
Good job, Aaron.
Thank you.